As we know, the nucleotide sequence of a gene is going to direct the amino acid sequence in that corresponding protein. We call this collinearity, or the fact that the order of the nucleotides in a gene is going to correlate directly with the order of amino acids in that corresponding polypeptide. So, for example, if you have a mutation at this particular locus here, the normal amino acid would be this, the substitution would be this, and we know the exact position of this. And the same with various different mutations. If you make a mutation at a particular location, you can make a pretty darn good guess on what's going to happen to that resulting protein, because you know what amino acid should be coded from that sequence. After the change, what amino acid will be coded at that particular sequence, and you know the exact location of that particular mutation. So, when you're going from DNA, remember that there's going to be two strands in transcription. You're going to have a non-template strand and a template strand. Okay? The template strand is going to be complementary to your mRNA. So in this case, imagine these two peeling apart. It's going to be read from a 5' to a 3' direction. Okay? So here is our 5' to 3' is going to be complementary here to the DNA that happens to be 3' to 5'. Okay? It's almost exactly the same as this non-template strand, because they're both complementary to the same thing. They're both complementary to this template strand. So anywhere a T would normally bind to an A, and that's indeed what we see here. A C would bind to a G, and exactly what we see here. Okay? So when we're looking at this mRNA, remember it's going to be read in groups of 3. Here we have codon 1, codon 2, codon 3. They're all going to be read in groups of 3. This mRNA, let's assume this is a mature RNA, is then going to be translated at the ribosome. Using the codon chart, you can look up and see that AGC is going to code for a serine. A GAC is going to code for an aspartate. And a CCC is going to code for proline. So, if someone gives you the DNA sequence for a particular gene, you can predict 100% accuracy what the protein sequence should be for that particular gene. And you can say with 100% certainty, if you get a mutation that, and say in this position here, it would go from, say, a serine to something else. Okay? So what I want you to do now, because the homework and the mastering genetics as well as the quiz are going to have some questions based on this, is just to practice. I will give you the template DNA. Try and figure out what is the complementary mRNA to this. What would the corresponding the corresponding amino acid sequence be for this? And then just think about what the corresponding tRNA is, or the anticodons for this particular sequence.